Gene Mutation Homeostasis, a presentation by Professor Gershom Zaitchik, Faculty of Medicine, Hebrew University of Jerusalem. In biology, a mutation is an alteration in the nucleic acid sequence of the genome of an organism, and mutations result from error during DNA or viral replications. This is a definition which we read in Wikipedia, but this definition cannot be accepted by homeostasis because in homeostasis there is no error. Mutation is a change and not an error. So here are the state of the two interpretation of homeostasis. We have one, the set of processes, and we are here in this interpretation is we are interested in homeostasis of processes, and these processes are maintaining homeostasis. But what we are interested here is that we want to know the homeostasis of the entire body or the upper process of the complex system, and therefore this is when we speak of homeostasis, we are speaking of the state of the homeostasis of the body. And this is actually a representation of milieu antérieur, which I discussed in the other presentation. So in this system, we study all what interests us in the body, and this is the homeostasis which I'm talking about. So homeostasis is a self-regulating complex system by which organism maintains stability and maximal optimality. So let's go back to mutation. This mutation which we are studying is affected by all processes in the body, more or less. But this is a reason why there is no error, because all processes contribute to this phenomenon, and the mutation is a phenomenon. So mutation is an indicator. Gene mutation indicates that gene has changed, and the change is function of the entire system. Mutation is a homeostatic or homeostasis state. It indicates an optimality change. So any things which we see in this homeostasis is either optimality, better optimality or less optimality, which I mean optimality change. So let's look what happened in other genes. Proto-oncogene is a normal gene that could become an oncogene due to mutation or increased expression. Oncogene is a gene that has the potential to cause cancer, and activated oncogene is already a gene in a state of cancer. So we look at two states. One state which is marked by the proto-oncogene. This is the indicator of the state. And the other state too is marked by the oncogene. And we remember that these oncogene and proto-oncogene are affected by processes of the body. And then we ask, is it possible to reverse the progression? We start with an oncogene and we reach the state 1 of proto-oncogene. And we know that it is impossible. But there are other ways to reach what we are looking for, because what we are interested in proto-oncogene and oncogene in order to delay cancer, not to produce cancer. And there are ways that you can delay cancer. And here's an example. promote processes that delay exit from proto-oncogene to state. So, which means that suppose you are here in the proto-oncogene state, which is a healthy state, and if you know how to activate processes in this healthy state that delay the exit from state 1, you reduced the probability of oncogene, the probability of cancer. And this I shall explain in other presentation. So, conclusion, more to come. Enjoy.